Hello students, in this video I am going to tell you the next topic, that is spermatogenesis. As you know, spermatogenesis is the formation of haploid sperms or spermatozoa from diploid spermatogonia. So, as we know that in human male the puberty starts at the age of 13 to 14 years and it continues without any interruption throughout the life, but the production is decline in latter life. So you know that inside the testis there is seminiferous tubules, which is the site of spermatogenesis. What is the actual site? That is seminiferous tubules. And the epithelium lining of the seminiferous tubule is called germinal epithelium. This all we have seen in earlier video, as germinal epithelium has two types of cells, germ cells and latter give rise to Sertoli cells. So I don't think that there is need of explanation of the function of these cells. Sertoli cells provide nutrition to the developing sperm, and germ cell function you all know. So let's come to the topic, that is spermatogenesis. Spermatogenesis occurs in four phases. First one is known as multiplication, then growth, then maturation, and then spermiogenesis. So the very first stage is multiplication, in which primary germ cells, which are cuboidal in outline or shape, they divide mitotically to form small spermatogonial cells, and at the time of sexual maturity spermogonial cells divide many times by mitosis to form large number of spermatogonia, which function as stem cells or mother spermatogonia. Spermatogonia. And in growth phase, you can say this is also called as spermatocytogenesis. Spermatocytogenesis. It is the phenomenon of formation of primary spermatocytes from the spermatogonia. So you can see here, this is the primary spermatocyte, which is again diploid in nature, and it is formed by the spermatogonia with the help of the process mitosis, and this comes under growth phase. Then next is maturation phase or formation of spermatids. So you can see here primary spermatocytes are large and diploid cells. They are having 44 plus XY chromosome, and they undergo meiosis first to produce small sized haploid secondary spermatocytes of two types, that is 22 plus X and 22 plus Y. So this is the very first meiosis which occur in primary spermatocytes. Secondary spermatocytes, however, possess replicated chromosome, and this having undergone crossing over. As you know, this is the speciality of the meiosis, and secondary spermatocytes divide by meiosis second to produce haploid spermatids with n number of chromosome and 1 n DNA. 1 n it represent the content of DNA. 1 n DNA. So as meiosis is a double division, as you know, the first one is reduction and the second one is equational division. So in reduction division you can see here that they all reduced to haploid cells. One primary spermatocytes or spermatogonium give rise to 4 spermatids. See here, see here, this is the 4 spermatids which is the product of meiosis second. And the last one, the last phase is known as differentiation phase. Differentiation phase, or this is also called as spermiogenesis. So it is a conversion of spermatids, see, spermatid into spermatozoa. For this spermatid must remain in contact with Sertoli cells. Do you know why? Because it provides the nutrition. So the spermatid must remain in contact with the Sertoli cells, which under the influence of follicle stimulating hormone, that is FSH of anterior pituitary, produces the required spermiogenic factor. And the different changes which occur during spermiogenesis are formation of acrosome, formation of acrosome. Acrosome, as I told you, that it is formed by the help of Golgi apparatus. Formation of acrosome, and then elongation and condensation of nucleus, as you know that the sperm nucleus is very much elongated and condensed. Then separation of centrioles and formation of axial filament, and development of mitochondrial spiral around upper part of axial filament, that is at the middle piece. So differentiation of a limiting membrane and formation of a flagellum, or which you can call as tail. So these are the differentiation or spermiogenesis which occur, that is formation of acrosome, elongation and condensation of nucleus, separation of centriole, formation of axial filament, and mitochondria is formed at the middle piece, and formation of flagellum or tail, and this produces a spermatozoon. There is another process which is known as spermeation. So there is a difference between spermeation and this spermiogenesis. So after the maturation of sperm or spermatozoa, it detaches from the Sertoli cells. Detachment from the Sertoli cell. So point to be remembered, spermiation spermiogenesis occurs when the cells are attached with Sertoli cells, and spermiation occurs when there is detachment of spermatozoon from the Sertoli cells. So the released so this it you can say it is the release of sperm upon its maturation. Spermiation is because it get detached from the Sertoli cells, so you can say that it is the release of sperm upon its maturation. So in this, sperms are detached from the Sertoli cells, and the sperms are fully mature at the time of spermiation. Fully mature, and there occur now physiological changes further. So this is all about the spermatogenesis. I think you all understand this process.